Shuka is an environmental scientist for the State Water Board, and she works in the statewide swamp program. She received her Bachelor of Science degree in Environmental Studies at California State University, Sacramento, and has nearly 10 years of experience at the state board working on a number of things, including ocean water quality policy, industrial stormwater permitting, and she's currently the statewide coordinator for the statewide bioassessment monitoring program. And before I hand off to Shuka, I don't know if you all are aware, but she is also the mastermind behind the scenes for CABW and has done an excellent job at getting everything together for this 2-day event. And thank you, Shuka. I can't express enough gratitude for that. And with that, I'm handing it off to you.
Thank you, Allie. I wanted to express everyone who has helped out put this event together. Just much thanks. Like it's been, it's a lot more difficult to set things up over Zoom, but hopefully we'll all be together in person next year. So I'm going to present our speakers for this session. First up is Chad Laughlin. So Chad is, he works as a senior environmental scientist for the state of California Water Quality Control Board, San Diego region. Chad oversees the water quality monitoring, assessment, and research program at the San Diego Regional Board, including the region's surface water ambient monitoring program. Chad is currently the project lead for the San Diego region on adoption of biological objectives for perennial and intermittent streams. So with that, Chad, I'm going to give things off to you.
There we go. All right. So today I'm very happy to talk to everyone about our efforts in the San Diego region on biological objectives. And so I work for the San Diego Regional Board. You can kind of see our area in the logo in the lower left-hand corner. I oversee our monitoring, assessment, and research unit, and our coverage area of the state includes San Diego County and portions of southwestern Riverside and southern Orange County, the coastal draining watersheds. All right, so our efforts on biological objectives really kind of predate 2013, being involved in a lot of bioassessment and whatnot. But in terms of the actual objectives themselves, they really kicked things off in 2013 as our regional board adopted a practical vision. And that practical vision specifically called out a couple of things for the board moving forward. One is to use latest science when taking actions and also for prioritizing actions, and really also to focus on environmental outcomes. And for those environmental outcomes, they actually specifically called out biological measures for ecosystems. So this really is, I guess, my presentation in one slide. So hopefully you'll listen after this one. But essentially, this is kind of a time frame for Clean Water Act up to our adoption, our regional board's adoption of biological objectives. So, back in 1972, the Clean Water Act was adopted, and literally the first line of the act says that the objective is to restore and maintain the chemical, physical, and biological integrity of the nation's waters. And up until the 1990s, most of that focus was really on chemistry-based regulation and technology-based regulation, controlling those types of sources and getting the technology in place to do so. And really the focus on chemistry was a natural first step for the Clean Water Act, but the modern Clean Water Act has really become a technology-based, chemically controlled pollutant law, and that was really never the intent. So in the early 1990s, US EPA started encouraging the states to adopt the biological objectives. And in response, the state of California really ramped up its biological assessment program, its bioassessment program within the early 90s, or early to mid 90s. And Region 9 started doing a lot of bioassessment as well. That bioassessment led to the development of a lot of regional indices of biotic integrity in the early and mid 2000s. There was a SoCal IBI, there was a Eastern Sierra IBI, and a hydropower IBI. And so all of those IBIs, I basically led to the need for a standardized IBI that would apply statewide. So, work was starting on that. And in 2016, we had our basin plan training review, which looks at potential new projects for our basin plan, and biological objectives was identified as a priority project. That's because at the same time, 2016, the new IBI, the California Stream Condition Index, or CSCI, as people have talked about before, was scientifically published. And so from that time period up, we'd been working on biological objectives, and the regional board, San Diego Regional Board, adopted the CSCI as an objective in December of 2020. So I don't expect everyone here is familiar with biological objectives or the regulatory process and how things work. So I thought I'd start out pretty simple from the beginning. From a Clean Water Act, really regulatory perspective, what we're talking about is water quality standards. And those are found in each region's regional boards and basin plan. And so water quality standards are where biological objectives fit in, and water quality standards themselves are actually made up of two things. They're basically beneficial uses for water bodies, and then water quality objectives themselves, so criteria. And I'll talk about those real quick for context. So beneficial uses it really comes out of the public trust doctrine. They're basically uses of water that need to be protected from a water quality perspective. And there's a variety of types of beneficial uses, including biological beneficial uses. And biological beneficial uses really focus on ecosystem integrity, that biological integrity, and they apply for a wide variety of water bodies in our region. Some examples shown here is intermittent stream, coastal wetlands, and then rocky subtidal kelp forest. There's also other beneficial uses that we protect with objectives related to things like drinking water, human water contact recreation, so swimming, surfing, scuba diving, that kind of thing, and fish and shellfish consumption. So to protect these beneficial uses, we have water quality objectives, and these are actually the criteria, narrative or numeric criteria, that really focuses on specific pollutants, both in the water column and also for sediment, and looking at water column and sediment chemistry and toxicity, and also the same thing for sediment, looking at toxicity and chemistry. However, there's really two main issues with using water quality objectives focused in this manner, and one is they're an indirect measurement of the beneficial use attainment for these biological beneficial uses, and two, there's issues with accuracy, things like when do you sample, what pollutants do you actually sample for, are the thresholds really appropriate or not? And so it's really been known for quite a while that biological beneficial uses need biological water quality objectives, and that's really because the organisms living in the environment are an excellent indicator of biological integrity and that beneficial use attainment. And like I said earlier, US EPA identified this as a priority in the early 90s. And so where we are across the United States is 38 states, including California's neighbors, Arizona and Oregon, have biological objectives. Some other regional boards have narrative forms of biological objectives. And I should also mention that back in 2003, the state of California and US EPA actually had entered into an MOU whose one of the major goals of which was to get biological objectives adopted for California. So really, how do you get a biological water quality objective put into our basin plan? Well, you have to do a basin plan amendment, and there are a number of legal requirements for that we have gone through or are going through. There's requirements under the California Water Code. There has to be a scientific peer review done, a SQL equivalency done, a review from the State Office of Administrative Law, and submittal to US EPA. There's also quite a bit of public participation that is required. So where we stand right now is Basin Plan Amendment was adopted by the San Diego Regional Board back into December of 2020, December 8th specifically. It included 3 components: narrative guidance for developing future biological objectives, which I'll touch on a little bit later, and actual numeric biological objectives for streams using the California Stream Condition Index, CSCI, and also a program of implementation, basically how the biological objectives will be implemented in the various regulatory and non-regulatory programs the regional board oversees. So why did we choose the CSCI, or California Stream Condition Index? We chose it because it's a standardized reference-based index. It's standardized for the entire state and our entire region. And it's also grounded in reference, scientifically defined reference. The 2 citations are here. Basically, the CSCI is an index of biotic integrity that uses benthic macroinvertebrates. I think a lot of the speakers have covered that so far. If you're acronym centric, you could say the CSCI is an IBI that uses BMI. But we really picked it because of the level of scientific rigor that went behind the CSCI's development and also defining the reference condition. So, so far we've gone through an extensive public process for adopting a biological objective for the CSCI. It started in 2016 with our triennial basin plan review. We released 2 drafts for public comments in 2018 and 2019. And in 2020, released proposed revisions to the 2019 draft for public comment. And then we had a board hearing in November of 2020, which was extended to December of 2020, where it was adopted. And throughout this process, we held multiple public workshops. We met with various dischargers and NGOs and gave presentations like the one today. I should also mention scientific peer review. So basin plan amendments are required to undergo scientific peer review, independent scientific peer review. That happened for this project. We had 4 peer reviewers. All 4 supported the project and approach, and they specifically supported using a reference approach for setting the objective. They really liked the inclusion of intermittent streams, and they liked the use of a modeled index, just what the CSCI is, and the percentile approach for setting the actual objective itself. So in regards to the public process, we did make a change in response to comments, quite a few changes, but one of the main changes of focus has been hardened stream beds. And so basically one of the changes we made from the 2019 to the 2020 revisions, which were adopted, was removing the applicability of the CSCI as an objective for hardened stream bed segments. The comments that we received really focused on these stream segments. And I want to be clear that the CSCI works in these stream segments and they can be restored. They've been restored throughout the San Diego region. However, we know that the concrete really must be removed from the stream bed for restorations purposes. There's some data from the Stormwater Monitoring Coalition on the left shown here that these stream systems just are not able to get a good CSCI score with the concrete in place, which is not really surprising. However, we still feel the CSCI is a reasonable objective to use to protect and restore these stream systems. That position hasn't changed. Basically, what we want to do is gather more information on the restoration timeframe for these stream segments, at which point we're going to come back to our board to either include them with a biological objective or potentially address these on a site-specific basis. So our next steps, since our regional board adopted back in December, the next step is to go to state board for consideration for adoption. The state board public comment period ended back on September 22nd, which means state board's got to respond to the comments and set a state board hearing date. That has not been done yet. So we don't know when a date for that hearing will be at this point. If state board adopts the objectives or approves the adoption by the San Diego Regional Board. Then it goes on to the Office of Administrative Law and then gets submitted to US EPA. So I thought it was pretty important, though, to not just focus on the CSEI and that objective. Really want to talk about our next steps. So I talked a little bit about revisiting hardened stream beds. We're going to kick off some information gathering efforts for the inclusion, looking at it, like I said, a site-specific basis, or if they should be done region-wide or watershed-based or something to that effect. Our board has also signaled that they want to add another biological objective for algae in streams using the Algal Stream Condition Index. And right now through SWAMP regional funds, we're funding the development of a molecular index for algae, basically a ASCI equivalent that's molecular based. And at the same time, we're using SWAMP funding and working with a lot of partners to develop indices for other priority habitats for those biological beneficial uses. We're looking at indices to basically monitor intermittent and ephemeral streams during their dry phase, basically when they're dry. And then we're also working on developing some level 3 monitoring tools and indicators for eelgrass beds. So that's really where we stand in Region 9, San Diego region today. So, but I wanted to be sure to thank the countless number of individuals and many agencies that really helped get to the point we are today, and who are continuing to help us in terms of adopting future biological objectives down the road.
Awesome. Thank you, Chad. As we wait for, we just got some questions. I had a question for you, but it's okay. So first question that we've received is, have you been able to define reference conditions for systems as spatially and temporally variable as your various temporary stream types?
Yeah, it's a good question. So for the streams that we have under the CSEI, the reference pool for those include intermittent and perennial streams that were capable of being sampled, basically developing a BMI community that could be sampled within a set index period. One of the challenges has been how to apply that reference, kind of the reference definition to ephemeral streams, because a lot of those have much smaller watersheds and have some processes that can impact them that aren't easily picked up by things like GIS. So we're kind of working on that right now.
Next question is, which beneficial uses are you associating the CSCI objective to?
It's a good question. I should have been more specific in that. So specific, like the two beneficial uses that the CSCI are really directly applicable to are the warm and cold beneficial uses in our basin plan. Those are really for stream ecosystems, and they specifically call out benthic invertebrates in them. However, we know that there's a lot of other beneficial uses related to biology that are dependent upon the BMI community and their conditions, things like rare threatened and endangered species like Southern California steelhead.
We have time for one more question. What additional information do you need to gather in order to develop CSCI objectives for hardened stream beds?
Good question. So really, we want to look at the time frame for restoration and mechanisms for restoration for removing the concrete from these hardened stream beds. And we know that they exist in a variety of watershed settings, up near the top of watersheds, down at the very bottom of watersheds, that can have an impact on that. So that's really the kind of information that we're looking at gathering before bringing back an objective or objectives. Don't really know if maybe it's CSCI, maybe it's the ASCI. That's the kind of information we're going to look into.
Great. Thank you so much, Chad, and thank you to everyone for your questions. I'm going to transition on to our next presenter, but Chad will be available to answer the remaining questions that have been submitted. So no worries. Thank you, Chad. Thanks. All right, so as our next speaker gets control of the slides, I'm going to introduce him. So our next speaker is Rafael Nazor. So Dr. Rafael Nazor is a freshwater biologist who specializes in freshwater bioassessment methods using benthic macroinvertebrates and algae. Working at large temporal and spatial scales, he has improved precision, accuracy, and sensitivity of bioassessments using the reference condition approach. In addition, he has studied the population genetics of rare and endangered invertebrates endemic to the vernal pools of California. His current research areas focus on the ecological impacts of hydrologic alteration, particularly in intermittent and ephemeral streams. He received his BA in neuroscience and behavior from Columbia University in 1999 and PhD in environmental science, policy, and management from the University of California, Berkeley in 2006. He joined SWIRP in 2006. So I'm pleased to introduce Rafi. Do you have control of the screens? The screen, one screen.
Excellent, thank you.
Good.
Thank you for that introduction, Shuka. And actually, thank you, Chad, for that's actually a really good setup for what I'm going to talk about today, which is to provide you guys with an update on some recently completed and ongoing studies on assessing biointegrity and biostimulatory conditions in modified channels. So Chad highlighted how this is, an important issue for his region, sort of establishing the breadth of applicability of biological objectives and what to do to manage these systems. And so I'm going to touch on some of the research that we're doing that will help his board make some of these decisions. And I also want to highlight my co-presenter, Scott Dusterhoff from San Francisco Estuary Institute. So as you guys have heard already several times, bioassessment indices such as the California Stream Condition Index, they're able to reflect the combined impact of all the stressors that are affecting a water body, in our case, a stream. And this is, like I said, this is one of the primary advantages for bioassessment, is the ability to get a holistic measure of the combined impacts of water quality, physical habitat, hydromodification, all in one measure. The flip side of that is that you can sometimes have a hard time picking apart the impacts of each of these individual stressors. So, and this is particularly true in modified channels, which can so pervasively and severely modify habitat conditions that it can be really difficult to see what's going on with our water quality. And that means that when we have programs that are intended to improve water quality, it's hard to know if they're working in these modified channels. So we need to have a better understanding of biological integrity in modified channels. And to go about this, we're trying to propose a number of classification systems that really speak to how biological integrity can be supported in modified channels. One thing Chad hinted at was that there are certain types of modifications, hard bottom channels, that need to be treated differently from other types. And that really kind of touches on what we're looking at. We need to have different sets of conversations about different channel types and really set appropriate management goals for our monitoring programs or our policy development. So how would we use these classes? One primary use is to identify classes of channels that are rarely meeting our standards, whether they're reference-based standards or other types of objectives. And you can, again, develop appropriate policies or management plans for each class. And in addition, set appropriate restoration goals for these different classes. Another use is to look for how biological integrity responds to stress in each of these classes. We can hopefully identify whether certain types are more responsive to water quality improvements than others, and therefore that can help us pick appropriate management actions that really will improve conditions in these systems. So it's sort of two different aspects, understanding the range of conditions and also understanding their responsiveness. So we've been talking about this issue for several years. Chad highlighted a handful of studies that the Stormwater Monitoring Coalition has done, but there's actually work done in other parts of the state. SFEI recently published a study on flood control channels in the Bay Area. I encourage you to check it out. And these studies have all really focused on bed and bank material as the primary way to describe channel modification. And that really appears to have sort of the most widespread support among watershed managers in terms of how to understand what channels are modified. There are, however, a number of different approaches you can take to attaining information on bed and bank material and that these studies have taken. The Stormwater Monitoring Coalition based their classifications primarily on site visits supplemented by photo interpretation. The SMC developed some simple checklists that really highlights some of the key features of modified channels to identify what types and where the modifications occurred. And this is really great because when you have this information, it's it's perfectly associated with your bioassessment data because it's at the same location. It's very high validity because you're actually observing the reach itself. But the drawback is that you can really only look at one site at a time. So while you can generate really great data sets this way, you're not going to get a map showing you where all your modified channels are. I'll contrast this with the approach taken in the Bay Area study, where they instead focused on shapefiles representing facilities that were constructed by the Stormwater Management Program up in Santa Clara County, as well as Sonoma County. And what they did is they crosswalked some of these inventories to categories that were similar to what the SMC had, focusing on whether the bottoms or the sides were hardened. And you can generate very beautiful maps that really give you a big picture view of what's going on in this watershed. The drawbacks are that you don't have perfect confidence that you've correctly characterized the site where your bioassessment data was located. You have categories like they call here mixed, which can represent some ambiguity as to what the actual channel looks like. And so there's a handful of drawbacks, but so ultimately you need a variety of approaches to really get the information you need to help manage modified channels in your watershed. The two studies are starting to show a more consensus about what these modified channels look like. As Chad mentioned before, the indices work. They are correctly showing us that conditions tend to be poor in our modified systems compared to natural systems. As Chad mentioned earlier, when you're looking at entirely hardened channels in Southern California, they were rarely, if ever, attaining reference-based thresholds. They were fairly consistently low scoring, and we see the same pattern in the Bay Area as well. Both Bay Area and Southern California studies show that the soft bottom channels represent a fairly diverse group. They tend to be mostly in poor shape, but there's a wide range, and we don't really have a good understanding of what's driving that variability. That's actually becoming a much more important issue, particularly for folks in the San Diego region, where these biological objectives do in fact apply. They're not really sure what to do about trying to attain it for those systems. In Southern California, we've gotten a really good idea of how these channels respond to stressors. But we're seeing is that in hardened systems, at least, most of the responses are fairly muted. So in these figures, you have hardened channels shown as blue dots and natural channels shown as red dots. And we're looking at a range of stressors on the x-axis. When you look at things like specific conductivity, natural channel scores decline as conductivity increases, and this is what you'd expect. But you really don't see much of a response in hardened channels. Conditions are fairly poor at a wide range of water quality conditions. And this is true for a wide variety of habitat or water quality parameters, although not all of them. For example, that last figure shows you how the scores respond to shading, and this could reflect the role of water temperature or provision of leaf litter for riparian vegetation as one way of potentially improving scores in hardened channels. We're also starting to see how context really affects things. And this is, we've barely scratched the surface on this in Southern California, but we have started to observe that there are a number of high scoring hard bottom channels, but they're all kind of the exceptions that prove the rule. So here's a stream way up high in the watershed of the Santa Clara River that receives discharges from the Drinkwater Reservoir. It's a fairly steep hardened channel that scores well. But the context is it's in an almost completely undeveloped landscape, as you can see in that photo there. So these are the circumstances in which hard bottom channels can sustain high CSCI scores. This may not be a very appropriate management goal for something like the lower San Diego River. The Bay Area actually got to look at this in a much more in-depth way. And what they found out is that imperviousness can affect condition even in hardened channels, watershed imperviousness. And you can see that in these red and orange dots. These are two classes of modified channels, the way that there's still actually an upward slope at that low end of the impervious area, in contrast to what you see in the green pattern, where you see a much more prolonged length of responsiveness. It sort of flattens out later. The Bay Area was also able to look at the role of channel maintenance, although those results, I would say, are a little bit less conclusive. They looked at a variety of typical maintenance activities like sediment removal, vegetation removal, or herbicide application, and both in hardened and softer natural channels. They don't really see a big difference in scores. However, this was held back a little by the fact that we couldn't really associate the time of the maintenance activity with the timing of sampling. We simply know that there's that type of activity occurring in those channels. So maybe new information might help us get some more insights here. So what are we working on now? I'll talk to you about some studies that have already been completed and are published. So there's actually a variety of studies by the State Water Board, the Central Valley Regional Water Quality Control Board, as well as the Stormwater Monitoring Coalition of Southern California. And a lot of these studies are focused on sort of confirming some of the patterns in one part of the state and other parts. We're basically trying to look at existing bioassessment data and see what we can find out about their channels and see if they uphold these patterns. And we're also strategizing new ways to actually get scores for streams in that lack definable watersheds, which is a particular widespread situation in the Central Valley, particularly in our canals down there. We're going to be expanding these studies to look at biostimulatory endpoints like ash-free dry mass or benthic chlorophyll A. And we're actually going to start focusing on management to improve conditions in modified channels. So how are we generating info on hundreds of sites with bioassessment data? We're going to be conducting site visits and field work, but we're also going to look at some related designations in various plans and documents, as well as start getting more use out of our PHAB data. And I think that's something that's probably going to be of interest to this group. So the Central Valley has an inland surface water plan that was drafted actually back in the 90s, and I don't think it's ever been formally approved, but it proposes a way of identifying modified channels by looking at data provided by stakeholders to the regional board, focusing on things like where the water is coming from and whether the channel's been straightened. And they have a bunch of water bodies classified in the appendix, like natural ag-dominated streams or constructed facilities. And when I look at the bioassessment data from those streams, and this is data from all of Region 5, not just the Central Valley floor, we're actually starting to see a pretty stark pattern. Similar to what we saw, modified streams are scoring lower than natural streams, although there really isn't a big impact of whether the water in those channels are return or supply. But what I think is most interesting is that when you look at a category of streams called constructed facilities, so those two panels on the right are both modified, but constructed ones are either artificial or implanted in historically dry washes. Those are scoring distinctly lower than other types of modified channels. So this is some new insights we're getting in the Central Valley. Physical habitat data would be great to use because we have so much of it, and we could then classify thousands of sites if we were able to use it. I'm focusing on Pebble Counts as well as our human influence measures as two ways of trying to infer the presence of modified channels. So our Pebble Counts, many of you know how we do this. We look at 21 transects, 5 locations in each transect, and we record the size of every pebble we encounter. Although if we encounter a pebble made of concrete, we record that. So the percent of concrete in the pebble count is an overall measure of armoring, at least on the bottom of the stream bed. This doesn't really tell us anything about soft bottom channels or earthen engineered channels, and it doesn't tell us about hardened channels where there's been a lot of sediment deposition on top. But we are seeing pretty good response where scores are highest as long as that's below about 10%. And that gives us an idea of how we can start classifying streams this way. We're also looking at human influence, where we look at what's going on in the riparian zones. And there's a few categories that suggest that there's been armoring, like walls and ripraps or buildings in the channel. And I basically looked at where these activities were occurring on the bank or in the channel. And I was able to calculate within a reach, what percent of it has armoring on one side, armoring on both sides, or no armoring at all. So I can sort of divide it up into these 3 compartments. And we again see a pretty good relationship with CSCI. These blue contours show that where the concentration of high scoring sites are, all above 0.79. And you can see that, of course, unarmored reaches where it's completely unarmored are where your high scores are most likely to occur. But there is a suggestion that armoring on one side can be tolerated to some extent more so than armoring on both sides. This is, I would say, preliminary. Putting the 2 pieces together, you're starting to get a good idea of what's going on channel bottom and sides. Although, like I said, this doesn't really capture what's going on with earthen engineered channels. So the next steps, we're going to do some field visits to confirm the accuracy of these classifications and sort of come up with some sort of finalized categories. And we're going to start calculating ranges of scores and biostimulatory patterns in each class. We're going to do the statewide as well as in regions where we have sufficient data, which is at least going to include Southern California, the Central Valley, and the Bay Area. We're going to also look at how both the CSCI and the algal index respond to biostimulatory stress within each class. The SMC in Southern California just approved of a new study to sort of extend these findings. And they're primarily focused on what can be done to improve conditions within existing channel forms by looking at not just water quality, but flow management and maintenance activities, as well as how we can improve conditions by restoring natural features. What's the timeline for improving those conditions? And with that, I'd like to thank you and take any questions you guys have.
Thank you, Rafi. As we're waiting for questions to come in, I was taking a look at that field data sheet that you showed a little bit earlier about how to classify or the documentation that you need to collect for channel standardization. I just wanted to know what your opinion is for like the best approach to standardize channel classification. Like, where would you start? Is there just a lot of, go ahead.
That's going to be like a key outcome of the studies that we're going to have some sort of simplified key features to focus on. I think it's not hard to tell when you have, a channel that looks like the one on the top, is it modified? That's not really going to be a challenge. It's really, how do you discern the earthen engineered channels from natural channels that might have had a lot of disturbance going on, but are still essentially natural? And I think we're going to try to focus on what are the key features to focus on there. Is it straightening? Is it, some sort of uniformity of shape that we're missing? And so I think we'll probably end up with key recommendations that can be added to our protocols for that.
Okay. Thank you. Looks like we actually did get some questions in, so I'll just read them out loud for you. What habitat types do you think are best suited for restoration based on your findings? Are hard bottom streams too altered to be good candidates?
I'm going to say that I don't think we know. I don't think we've done enough restoration activity, and even if we have, we've not done enough research on them to have a sense of what's most likely to work. So I think, those are great questions. I think we do know that restorations do need to take a watershed scale perspective and can't just focus on fixing one reach at a time. Those are unlikely to succeed.
Okay, and the next question that we have is, upcountry ranchers use BMPs like livestocking and willow recruitment to restore stream banks. Is that deemed a modification?
That's a good question. So I would say I've been using the definition of modification as being a deliberate change of the channel morphology. So I'm not talking about, incision that might occur that wasn't a deliberate intentional change. There's probably a lot of gray areas, and I think what you're talking about fits into that, but I would say that's an activity that is not intended to alter the channel form, so I probably would not consider it, but I suppose there are cases that could vary.
Okay. Thank you. I don't know if we're going to get any more questions, but if you do submit questions or, I'm so sorry, my video is not on. Sorry about that. But if you do have questions or you're in the middle of typing it, go ahead and submit your questions. I hope Rafi will be here available he can type answer like his responses to you. So just go ahead and submit any questions that you have. So that's actually it for this short session. If you have any additional questions for our presenters today or any of our presenters today, you can either email the SWAMP email inbox, or you can email me to get contact information for any of our presenters. Wanted to thank both Chad and Rafi for presenting today. Thank you so much.
Thanks again to all the presenters today. There were some really great talks, excellent questions, and good discussion around those questions. And like we've mentioned throughout the day, if you'd like to follow up with any of the speakers, feel free to reach out to them if they've made their contact information available. Otherwise, you can always reach out to that swamp@waterboards.ca.gov email, or to either myself or Shuka, and we can connect you as well. I just wanted to quickly I threw this slide up earlier today. I'm not sure if you all can see that very well, but we do have a project going on. And Peter, I don't know if you're on, if you wanted to say some things, I'm happy to.
I am on.
Yay. Hi, Peter.
Hi.
So I'll hand it over to you if you'd like to talk to everybody about these EDNA kits.
Yeah. Hi, everybody. I'm Peter Haupt. I'm with the Surface Water Ambient Monitoring Program, otherwise known as SWAMP. We're searching for partners that would like to participate in an environmental DNA pilot project. As you've heard, eDNA is a new way of examining aquatic ecosystems, and sampling is super easy. Just filled out a field collection form on your phone and push some water with a syringe through a filter, send that off in the mail, and all of the necessary equipment, including a postage paid envelope, is provided by SWAMP free of charge. In return for sample collection, the SWAMP eDNA Metabarcoding Monitoring and Analysis Project, CMAP, will provide analysis and support in the form of direct communication with the project leads, a web portal that allows users to look at the map, click on their collection site installations are occupying their water. Knowing this information is useful in myriad ways, and CMAP scientists are always available to answer questions and help participants analyze their sampling results. So we're seeking partners from a broad range of backgrounds, locations, and skill sets. One of the challenges of this new way of collecting data is to automate the field forms, make them easy and user-friendly while allowing us to receive information of known quality. So SWAMP is searching for tribal and community science partners, educational partners that would like to participate in the eDNA sampling project. For the past 2 years, we've been collecting phytoplankton and fish results, but this year we will expand to include macroinvertebrates as well. So there are 2 ways to participate: passively by joining our email list and keep up to date on what CMAP is up to. The other way is by actively participating with us and becoming a member and collecting samples. There are a couple of forms to fill out that are linked in the chat. And that's my pitch. I hope you join up. Thank you.
Awesome. Thanks, Peter. And there's actually a question for you if you want to hold on, which is it possible to participate outside of USA, out of America?
No, we're going to keep it in California. That's one of our goals, is to show how we can expand our footprint statewide. So that's really, it's subject to California only.
Cool. Well, thanks again, Peter. And all this information is here, folks. So I'll keep this up. So you still have access to the email list and requests for free kits. And I think with that, yeah, we will see you all tomorrow. I want to, again, just express sincere gratitude to all the speakers and participants, and for another great CABW day 1. and be back here tomorrow for lots of great talks. Thanks, everybody.